She has a PhD from Indiana University, 1995. She has monitored air quality in the places as far away as Beijing, China. And I caught this quote from Oregon's Agricultural Magazine, Oregon's Agricultural Progress Magazine in 2009, in which Staci said of her trip to Beijing, it was the most polluted place I had ever been, and the filters, the air filters, were the dirtiest I had seen in 20 years of air sampling. So I'm sure she's glad to be living in Oregon instead of Beijing. But today, Dr. Simonich leads a research team that works through the NIH-funded Superfund Research Center to understand how people in the United States and in China are exposed to polycyclic aromatic hydrocarbons, or PAHs. And I understand that when she's not in her lab or not at home taking care and working with her family, she is out on the coast on a surfboard riding waves. So maybe she'll tell us a little bit about that too. Please give a warm welcome to Staci Simonich.
Good evening, everyone. It's really nice to be here. It's surprising to see so many people, but I feel really honored to have many of my students here, many of my friends here, many of my colleagues here who I've worked with. So also my children, my friends, I really appreciate it. So I think part of what I'm feeling right now is this idea that what goes around comes around, that you get back what you give. And that's certainly what I'm feeling tonight by having all of you here. So in the atmosphere, there's also this concept, in my studying it 11 years now here at Oregon State University, that when a pollutant is emitted to the atmosphere, that if it's long enough lived in the atmosphere, that it actually can move long distances and in theory could circle the globe and come back to you. So this idea of what goes around comes around. And my research group is definitely known for chasing air pollution all over the western US, and I'll tell you about that tonight. I often say I have the best job in the world, and I think you're gonna see that in part tonight. So let's start here. When I arrived at Oregon State University 11 years ago, one of the first things I did was to have someone draw a cartoon of what I saw the vision of my laboratory as being. And so I had this cartoon drawn, and you can imagine on the left-hand side that this is Asia, kind of an amorphous blob here, not to show any definite boundaries, but Asia here on the left-hand side, the western US on the right-hand side, and the idea that you could have air pollutants moving across the Pacific Ocean, and in the spring of the year, this transport time is as short as 5 to 7 days. So this idea that pollutants could move from Asia to the western US, end up in our valleys and even in our mountain ranges, but also the idea that we have our own regional air pollutants here in the US. Certainly, we're in the valley, we have I-5 going by, we have manufacturing, we have agriculture. We certainly have our own sources within the western US, and that these pollutants could actually also be transported to mountains. And so in my talk tonight, I'm gonna first tell you about atmospheric transport and how that moves and what we receive in the western US. And then the second half of my talk, I'm going to talk about deposition to mountains in the western US. And this idea that you could actually have increasing concentrations of pollutants with colder temperatures and a concentrating effect of air pollutants in mountains. So my laboratory does study semi-volatile organic compounds. So what is an organic compound? Grace, what's an organic compound? So it's not a chemical you go to the co-op and buy. It's not an organic chemical from that standpoint. They're compounds that contain carbon. And we study in my laboratory semi-volatile organic compounds, compounds that are volatile enough to go into the air and be transported long distances, and non-volatile enough to come out of the atmosphere in rain and in snow, and maybe even remote locations like the tops of mountains in the Arctic. So semi-volatile organic compounds, the ones we study tend to be persistent in the environment, meaning that they don't degrade very quickly in the environment, either with biology or with other forms of abiotic chemistry. And many times the compounds we study contain carbon-chlorine bonds, for example, polychlorinated biphenyls. That carbon-chlorine bond is quite strong, so they're resistant to degradation in the environment. Also, many pesticides, both current use and some, well, mostly historic use, but some current use pesticides also have carbon-chlorine bonds. And we also study organic compounds that are bonded, have a carbon-bromine bond, and some of these are the brominated flame retardants, polybrominated diphenyl ethers. And you're not sitting on cushions tonight, so I often like to freak the audience out a little bit by telling them they're sitting on these. But in this case tonight, we're not sitting on cushions, but they're often found in furniture and in the foam that is in furniture. So polybrominated diphenyl ethers, lots of different pesticides we study. Some are historic use, meaning that they've been banned in the US for decades, maybe still used in other parts of the country, could be currently used in the US, a series of polychlorinated biphenyls that were used historically in transformers, no longer manufactured in the US after about 1979. And then polycyclic aromatic hydrocarbons. These are products of incomplete combustion. So any form of combustion, whether it's a forest fire, our cars, diesel engines, produce these PAHs, some of which are shown to cause cancer or be linked to cancer. So a wide variety of different chemicals that we study from different sources. So products of incomplete combustion from combustion sources, polychlorinated biphenyls from urban areas or industrial areas, and lots of different pesticides as markers of agricultural areas. So by studying this big list of chemicals, we can tell something about different sources, where they're coming from, different source regions. The other thing about this list is that they have different properties. Some are lipid soluble, some like DDT, classic example of accumulation in organisms. Others are more water soluble, less likely to accumulate in organisms. Some are more persistent in the environment, could have half-lives of months or even a year in the environment. Some, such as gas phase PAHs in air, only persistent on the matter of hours. So by mixing together this suite of different chemicals, we can tell a lot about an air mass and where it came from. So my students spend a lot of time developing analytical methods to measure this wide list of chemicals in different media in the environment. And we often have to make measures at very low levels. For example, 10 to the minus 9 grams. That's 1/1000th of a gram. It's also 1/1000000000th of a pound. So that's the type of level we can measure at if we're going to remote locations that might have low levels. Of course, if we're working in China, then we don't need quite those low levels, so it just depends. So let's start here in the atmospheric transport side of the first part of my talk tonight. And we've done a lot of work in my laboratory in China. We've been working in China for about 6 or 7 years now, and in fact, did a lot of work around the Beijing Olympics. Some of it was not always popular with the Chinese government, but I think it was pretty important work. We did studies on the outflow of air pollution from different Asian countries. Okinawa, Japan is about a 1-day transport time from different Asian countries, so we could sort out with the chemistry and the meteorology which pollutants were coming from which countries. So we've spent a lot of time working on the left-hand side of the Pacific Ocean, but tonight I'm going to focus in here on the right-hand side of the Pacific Ocean, more close to home in the western US. I'm going to focus on these 3 sites that we've worked at over time, one on the Olympic Peninsula in Washington at 500 meters, one Mary's Peak just outside of town at 1,250 meters, and then at our site at Mount Bachelor at 2,700 meters. So we're at some pretty remote locations in the Pacific Northwest at different elevations so that we can characterize the atmosphere at those different elevations and how it might be similar or different. So let's start here at Mount Bachelor. I can tell you a little bit about our story there. And yes, I like to ski, but that's not the only reason we're at Mount Bachelor. And actually, it's become a very important site, not just for my laboratory, but for laboratories at the University of Washington, and actually also for several other laboratories throughout the US. So it's kind of become a hot spot for studying air pollution, especially trans-Pacific transport from Asia over time. So if you've been to Mount Bachelor, you know it's beautiful. You know there's the summit building at the top. It's in the Cascade Range. It's remote. It's high in elevation. One of the best things is that there's power at the top because of this lift building. And you can also just take the lift right up and jump off and run the air samplers, drawing air in from outside. So it's a very good site from that standpoint. So one thing we do is we have to mix the chemistry with the meteorology and figure out where an air mass came from. And so we use meteorological models to try and backtrack in time where an air mass came from when we sampled it here at Mount Bachelor. And so when you look at where the different source regions are in the western US, if you're looking at people, where do people live? Well, they live in Seattle, they live in Portland, they live in various places in California. We can calculate from a spot in time with our models for Mount Bachelor if those air masses passed through these different source regions, either agricultural or urban areas in the Pacific Northwest and in the western US in general. We can also calculate back in time if that air mass came out of regions in Asia, including China, also passing over Japan and Korea. So we can put our measurements together with this modeling and calculate the percent of time an air mass spent in these different boxes to try to sort out where different pollutants came from. And so this is an example. I'm going to show you more about this one day. It was in April, and it was a very strong trans-Pacific transport day. This transport time was about 5 days. Other times of the year, for example, in summer, we see that the air masses are mainly drawn off of the Pacific Ocean and regionally. So we can begin to look at concentrations of different pollutants. These are those polycyclic aromatic hydrocarbons, the ones that were funded at OSU from NIH in the Superfund research program to study, some of which of these are mutagenic. Some of them are in the gas phase in the atmosphere, and some are on very fine particles in the atmosphere, so 2.5 micrometers or less. And those are the ones that we're most concerned about because those are the ones that you can breathe in. The particles can be trapped in your fine lungs, fine areas of your lungs, and not expelled when you cough. So essentially, if you breathe in a very fine particle with PAHs on it, and it's lodged in your lungs, you'll be exposed to that mutagenic chemical for long periods. So this is data from Mount Bachelor. These are the gas phase PAH concentrations in red. That's the red line shown here. The black are the particulate phase PAH concentrations shown in the black line. And these are different time periods that we collected samples and studied at Mount Bachelor. And this spring Intex B was actually a big study, and NOAA actually flew one of their airplanes for air pollution measurements right by Mount Bachelor as part of the study and part of our measurements. And so what we found is that these PAHs should be coming from same sources. They should be coming from automobiles, they should be coming from other forms of combustion, and in theory, these two should be linked. And if you just look at the patterns in red and the patterns in black, they don't look very linked. And if they were coming from the same places, they would be linked. And we can calculate correlations to see if they're correlated. In fact, these two forms of PAHs are not correlated with each other, which was kind of curious. It told us something was different about them and where they were coming from. But then we looked and compared with our long list of chemicals I showed you in my third slide to see where they could be coming from. And we linked them to different markers of urban areas, or rutene and levoglucosan. These are markers for forest fires, for wood combustion. We saw that these gas phase PAHs were linked to either urban pollutants or to forest fires. The particle phase PAHs weren't linked to these markers for urban areas or forest fires. So we started to put a picture together, and in fact investigated more this day where we had high, it was in spring of 2004, it was April, and we saw high particulate phase PAH concentrations but low gas phase PAH concentrations. And in fact, that was that day that we saw very fast and rapid trans-Pacific transport. So we've done additional studies to show that significant amounts of these very fine particles can be transported all the way out of Asia, up high in the atmosphere, and can arrive here in the western US. So it's definitely an episodic event happening more in the spring of the year, but it's part of what we receive here in Oregon. So we can start to put together with statistics and with our modeling the percent of time an air mass spent in a given region and connect that to the different pollutants we study. The gas phase, PAH is being linked to more to sources in Oregon, and these particle phase, PAH is being linked more to sources in Asia. So that's the type of thing we can start to do to interpret our data from the atmosphere. So the other important role that we've discovered over time in the past 4 years or so is the importance of forest fires. And we have a couple markers for that. One of them is levoglucosan. It's a marker for wood combustion. It's actually produced from the combustion of cellulose. That's in red on this graph. Again, this is data from Mount Bachelor, measured over time, different seasons. Retine is a marker for softwood combustion. So when you combust pine, you would produce retine. So we can look at when we measure high concentrations of those, and they are linked to forest fires. This is a satellite image. These red dots are fire starts, places where fires were burning in the western US. This gray is the smoke emitted from those fires. And this is our air mass back trajectory in time from the sample we collected here in the summer of 2004. The air mass passing through smoky regions, picking up these markers of combustion. Other examples here, more local fires around Mount Bachelor that we detect, and then another example here. So maybe it's not unusual that we would measure markers of wood combustion from these forest fires when our air mass pass by them. But what was somewhat unusual is that we started to see that the concentrations of other pollutants were also higher in air masses that passed through forested regions. In fact, polychlorinated biphenyls were positively correlated with these markers for forest fires. And we looked at days where we measured high PCB concentrations or high-ish PCB concentrations out Mount Bachelor. We saw that those were the days when they were also linked to forest fires. Why would forest fires be producing PCBs? Why would PCBs be emitted from forest fires? Strange. Okay, I'm going to tell you more about the story as we go along. So now let's move to two of our other sites we've used over time, Chica Peak Observatory and Mary's Peak. Everybody around here knows Mary's Peak. And so during a summer, we collected samples simultaneously at both sites at these different elevations to study how large of an area would be affected by trans-Pacific transport and how it might vary with elevation. So we did these studies in the summer of 2003. It just so happened that during that summer, millions of hectares of land in Siberia burned. And so this is a satellite image of the Earth. These are the fire starts in red that are burning in areas around the world. And what you don't see in this image is large amounts of smoke being blown from west to east. And so we can overlay the satellite image of all these areas burning with our air samples, the purple one with their air mass back trajectories for Mary's Peak, and the orange being the air mass back trajectories for Chica Peak on the Olympic Peninsula. And so we measured high PAH concentrations from those forest fires. So evidence that not only are we receiving maybe PAHs from urban areas in Asia, including in China, but when there's large fires burning, that we also pick up those emissions as they're transported across the Pacific Ocean. So we saw high PAH concentrations, some of them higher at Mary's Peak, others higher at Chica Peak. But the surprising part of it was that we measured high concentrations of different pesticides also in those air masses. So like we saw at Mount Bachelor, we saw emissions of PCBs and pesticides, higher concentrations during forest fires. We saw higher concentrations during when these large areas of Siberia had burned. Again, showing us that somehow forest fires seem to be emitting PCBs and pesticides. So raise your hand if you remember the BNB Complex Fire. So, and we often drive through that area, right, when we drive up into the Cascades. So that same summer, we had the BNB Complex Fire burning, and we were collecting samples at Mary's Peak. And this is an image, a satellite photo showing that fire burning where Corvallis is located. And we measured higher PAH concentrations at Mary's Peak during that fire. So again, a regional fire showing evidence of these PAHs, but also measuring higher concentrations of some pesticides during that fire also. So we had kind of 3 lines of evidence coming together saying that when forest fires burn, they seem to release these man-made chemicals. So I had a very clever student, and she's now working for the FDA in Washington, D.C. And so she went up into the Cascade Range, into the area of the BNB Complex Fire, and she sampled soil all over the burned area. She sampled soil all over the unburned area, composited those samples, and then took them back to the lab and measured those soil samples for the big list of chemicals I showed you, the burned ones, and the pesticides, and the PCBs. And so what we found is that the green bar is the concentration of the different pesticides and the different PCBs, and the unburned soils, the red, were the concentrations in the burned soils. So in fact, the burned soils in the same location had 75 to 95% lower concentrations than the unburned soil. So how did these PCBs and pesticides get into the forests? Well, I'll be telling you a little bit in a few slides about the national parks, and some pesticides might be applied to some of these forests in some areas, but not such a wide range of pesticides. And in fact, the plants growing in forests are very efficient at cleaning the atmosphere. They have lipid surfaces on the conifer needles, the lichens accumulate them. There's a lot of cleaning of the atmosphere that goes on in forests, one of the great things about forests. And as those needles fall to the ground, they begin to accumulate in the soils. Also snow. I'm going to show you some data for snow in a minute. Snow falls, rain falls, and these organic pollutants bind to the soil particles and stay there. So that when a fire comes through the area, it actually re-releases some of these pesticides and PCBs to the atmosphere. Now hopefully some are degraded along the way, but some are definitely released. It's definitely allergy season. So where are we going with this? Well, this is my student An. She's from Thailand. And what we're interested in right now is the chemical reactions that can occur in transit to the US West Coast. So we've taken particulate matter from Beijing, and so this is actually a little bit from my introduction, collected air samples over time in Beijing. This is a 24 hours of sucking Beijing air through filters. The white is the unsampled part of the filter. The grayish black is in fact the particulate matter that's poured through the filter and sampled. And so we cut these filters into pieces. We react half, we don't react half, with different chemical oxidants from the atmosphere. We do this at UC Riverside in a very famous atmospheric chemist laboratory. And An measures the difference between the unreacted and the reacted side of the filters in these big, big chambers. And so we're trying to figure out if the chemical reactions that occur in transit to the US West Coast, if they in fact produce more toxic pollutants in transit as they react. So we do some mutagenicity testing on the filters. We also do the chemical analysis on the filters. So that's where we're heading in our atmospheric transport side of my laboratory. So the second half of my talk is about atmospheric deposition. And this was the Western Airborne Contaminants Assessment Project. It really was a project of a lifetime. And in fact, there's at least 3 scientists in the room that participated in that project. Linda Geiser from the Forest Service, Dixon Landers from the US EPA, and Marilyn Irway, I think I saw her, from the US EPA. So this was, I get goosebumps when I talk about it. because, who else, Linda? Jill, oh yes, Jill. How could I forget Jill? Yeah. Jill, who works in my laboratory, this was part of her master's thesis. So really it was a project of a lifetime. It was the Forest Service, the U.S. Geological Survey, OSU, EPA, anybody I forgot, National Park Service. So all coming together, a team of about 20 scientists coming together, a bunch of students going out into national parks and trying to figure out determining if these persistent organic pollutants, same thing as semi-volatile organic compounds, if they're present in the parks, how did they get there, what are the sources, how are they distributed, how do they move in the ecosystem, and what impact do they have? So really just an amazing project. And so when you think about it, 1/3 of the US is covered in mountains. You know, I grew up in Green Bay, Wisconsin. You can probably hear that in my voice. We have other guests from Wisconsin here tonight. We don't think about mountains much. We don't do a lot of alpine skiing. We mostly drink beer. But, but in Oregon, you have good beer and you have mountains. So, okay, so 1/3 of the US is covered in mountains. The value of that in terms of recreation, in terms of the water we get from those mountains is incredible. It's a huge resource, really important. So this focus of this project was on these high elevation ecosystems, and also you're gonna see high Arctic ecosystems or Arctic ecosystems, trying to figure out if pollutants were there and how they got there. So how could an organic pollutant move to mountains? How could they get there? Well, if we have source regions, let's say we have the San Joaquin Valley, or even our valley, where there's agriculture and there's cars driving, you can have this grasshopper effect with the cooling, or let's say the heating of during the day, volatilization from soils, and then deposition at night when it's cooler. So essentially a grasshopper effect where over time, different pollutants could move up the side of a mountain and accumulate at the top or somewhere along this range, depending on its vapor pressure, how volatile it was. So that's one way that pollutants can get to the top of a mountain. Also, like we use Mount Bachelor for, we're up there to trap and intercept these global winds. Figure out if we're getting a global wind or a regional wind. So global winds carrying pollutants from far off can be intercepted at the top of these mountains. We can also have a conveyor belt effect. Again, if we're near source regions, either cities or agriculture, we can have upsloping during the day, warming up in the valley, pollutants move up, pollutants stick because of their semi-volatile nature, air mass comes down at night, upslope, downslope effect. And so literally a pumping of pollutants up the side of a mountain near source regions. So those are some of the ways the pollutants can get up there. And so we set out to work in over 20 different national parks, up from above the Arctic Circle down to near in Texas and Sequoia National Park. These red parks were the parks that we did very detailed studies in. I'm going to show you a slide on that in a minute. And these blue parks are parks that we did smaller scale studies in. So you know, working in some of the most amazing places, certainly in the country, if not the world. But I didn't get to go anywhere. I stayed home. That's okay. So a great team of scientists going out and collecting these samples. So the studies the samples were collected between 2003 and 2005. My laboratory focused on the organics measurement, that big long list I showed you in that third slide. And so think of this as transport from long range versus regional, and lake catchments that were perched in these parks in different select spots. Deposition coming in the form of snow, also rain, but these are cold ecosystems, so a big part of what's coming in terms of deposition is snow. So we needed to be able to make measurements in snow, and for example, my laboratory made measurements. We'd go out and collect 50 kilogram samples of snow. That's like 122 pounds of snow for a sample. Bring it home. They were like giant turkeys, melt them, and measure the pollutants inside of the snow. Other measurements we made, we measured the fish in the lakes to see what they had accumulated in terms of pollutants. We actually had this nifty device where we could put a submersible pump within the lake and do the water extraction, 50-liter water extraction, right in the lake and bring home the cartridge that the water had passed through and measure that. So we didn't have to haul back all these water samples. And then lake sediments were very useful because Dixon could collect a sediment core from the lake and some of the deepest parts of the lake, and we could slice them. Marilyn could slice them and date them and come up with kind of like a tree ring, but a sedimentation layer that we could put a date on to see if the different pollutants were increasing or decreasing over time. And Jill, as part of her master's thesis, used both conifer needles and lichen with Linda Geiser and used those as passive air samplers of the atmosphere. So unlike Mount Bachelor, no power, right? We can't set up an air sampler in these locations, but we can go in and we can collect different parts of the ecosystem, develop new analytical methods to measure them at low levels, and then trace how they might move through that ecosystem. So that's what I'm going to walk you through now. So this is the annual snowpack for the different parks: Sequoia National Park, Rocky, Olympic, Rainier, Glacier, Denali, Noatak, and Gates. So we're up in Alaska and the Arctic here. You know, we're in some pretty agriculturalized areas with Sequoia and Rocky and Glacier. Olympic, it's probably the furthest out, yes, in the west, in the Pacific Northwest. Rainier, more inland. And so these are the different years we collected snow. The black bar is 03, the gray bar is 04, the white bar is 05. And on this x-axis, the left-hand side are current use pesticides. These are current use pesticides. The right-hand side is historic use pesticides. So what are some of the things you notice about this? Well, one thing is that these historic use pesticides, which have been banned for decades, are still coming into the park. You know, this isn't a glacier. This is annual snowpack at the end of the snow accumulation period. So first of all, I thought we banned these guys. Why are they still coming in? Well, and then these current use pesticides, they're certainly coming in, and in fact their concentrations are generally higher than the historic use pesticides. The other thing we looked at was, well, year to year, this profile of which pesticides were coming in at the highest amounts in the parks was pretty consistent. Now let's look at Sequoia. Okay, Sequoia National Park, dacthal, the current use pesticide dacthal, made the largest contribution of pesticides coming in. And for all 3 years, that was true. So the profiles among the different parks are very different. Each park has a different sources of pollution to that park. So that each park looks different, but year to year, in general, within that park, it's pretty consistent. The concentrations might vary slightly over time, depending on how much snow they receive that year and other factors, but year to year, it was pretty consistent as to what was coming into an individual park. So that was pretty interesting. And even in Gates of the Arctic, Noatak, Denali, we saw pesticides, including historic use pesticides, coming into those parks. So we tried to figure out why. You know, were these historic use pesticides coming from long range, from sources far away, or were they our own sources? And so for each park, we calculated how much agriculture was around that park within a 150 kilometer radius of that park. And we chose that distance because that's about the distance an air mass can travel in about 1 day. So within about a 1 day's transport time, how much agriculture was around that park. And so a little bit of a surprise to me was that of our parks, Glacier had the highest % agriculture around that park. And a big part of that is actually the canola growing above Glacier National Park in Canada. So Glacier has a lot of agriculture. Sequoia, of course, has a lot of agriculture from the San Joaquin Valley. Rocky, about third in that. So Glacier, Sequoia, Rocky, some of those parks with the greatest contribution from agriculture. What about Alaska? Is there agriculture in Alaska? Not much. Not a lot. But, you know, maybe a little bit, maybe some pesticide use there. But for our calculations and for our rough calculations we were doing, we assumed that there was no agriculture there. So we tried to relate these numbers we calculated in terms of percent agriculture around a park with the pesticide concentrations in the park. And so this is an example of that. This is the pesticide endosulfan. And we plotted the different concentrations that we measured in snow against the percent cropland around that park. So these are the Alaska parks with, we assumed, 0% cropland moving up into Glacier and Rocky and Sequoia. So we assumed that this baseline level in Alaska was due to long-range transport. And anything above that was due to regional transport from the US. So for a given pesticide, we could calculate the percent of what we found that was due to long-range transport, the red number, and the percent that was due to regional transport, the blue number. And so the first thing this told us was, of course, those regional sources were important. And in fact, they were also important for historic use pesticides. So why would historic use pesticides be linked to the percent of agriculture around a US national park? Weren't they banned? Well, the whole reason they were banned is because they were persistent. So even though they were used decades ago in the croplands around the national parks, they still persist in the soils. You can still go out and measure them. So we still have a cycle of those farmlands being used and being plowed, and those pollutants revolatilizing into the air, moving into the parks and being deposited. So we were able to calculate the percent contribution from either long range or the regional. So certainly regional sources played an important role. And in fact, some of the data that we generated in WAKE and WAKEAP with regard to endosulfan was brought to the EPA from the National Park Service and has resulted in some restrictions in endosulfan. Okay, let's talk about the role of topographic barriers. This is Rocky Mountain National Park. This yellow line is the Continental Divide. Here's the west side. Here's the east side. Where do all the people live? They live on the east side. 98% of the population is on the east side, and all the agriculture is on the east side also. 90%, 92% of the cropland. So here you have the steep Continental Divide. Not a lot of people, not a lot of agriculture, lots of people, lots of agriculture. So it was a pretty sweet experiment when we started to think about where are the winds coming from. Okay, they're coming from the west. You could have this conveyor belt effect that I discussed early on, where you could be concentrating pollutants on either side, and the two sides might be different if this topographic barrier played a role. So we chose two lakes, one on the west side, Lone Pine Lake at 3,024 meters. We chose one on the east side, Mills Lake, at 3,000 meters. This is the lake bathymetry. These stars are some of the deepest part of the lakes are shown in dark blue, and where Dixon Landers, who's sitting in the back of the room, collected the sediment cores out of those spots with the stars. And so they're only separated by about 10 kilometers as the crow flies. But in fact, they're separated by a peak, the Continental Divide, which is about 1,500 meters from the position of the two lakes. So similar lakes in a lot of ways, similar elevations, separated by this divide, one on the east, one on the west. And so these sediment cores were very useful to us for interpreting what was happening on either side. And so that's what's in this slide. This is that current use pesticide endosulfan. Here's the west side, that Lone Pine Lake, and Maryland sectioned off all those different layers of the sediment core, and we were able to date them with cesium-137 and lead-210 and assign different dates to the different layers. And then go ahead and measure the endosulfan concentration or flux in those different layers. So same thing on the right-hand side at Mills Lake. You remember where all the agriculture is. So these scales are the same on the two x-axes. The first thing you notice is that the concentrations are much higher on the east side. We can also see that the concentrations have increased over time. You know, this is current day. This was the top of the sediment. And as we went back in time, this is where US registration was. And we have some low concentrations, could be some smearing within the sediment core to those lower times, but certainly increasing with time. So we can calculate doubling times, kind of the inverse of half-life, for this pesticide, current use pesticide within these lakes. So we see that the concentrations or flux are much higher on the east side with a faster doubling time. So certainly a pesticide that's coming into parks currently. So what about something that we've banned in the US? Technical chlordane, banned in, I think, 1988. Not that long ago. Used as a termiticide. Again, the west side lake, the east side lake, the sediment cores, the layers. Here's where US registration occurred. Here's where US restriction occurred. And so on the west side, we see somewhat of a statistical decrease in concentrations. On the right side, we don't see a statistical decrease in concentrations. And again, we see much higher concentrations or flux on the east side. What about DDTs, our old friend DDT? Historic use pesticide, here's where it was registered in time, here's where it was banned in about 1973. And we see a decrease on the west side, but not much of a decrease on the east side. But again, higher concentrations on the east side. What about those markers of incomplete combustion from urban areas, from people driving their car, from population essentially? We again measure higher concentrations on the east side, and this doubling time is almost the same as the doubling time in the population on the east side. Where on the west side, we see lower concentrations and we see a decrease in time. We can actually measure a half-life. So this appears to be directly linked to population increases on the east side, where the others appear to be linked to agriculture on the east side. One last one, these are the polybrominated diphenyl ether flame retardants. On the west side, these would also be linked to people, where people are consumer products and are the flame retardants. So on the west side, barely above the detection limit. On the east side, measurable concentrations, certainly higher than the west side, and the doubling time we measured in Mills Lake sediment is the same as the doubling time that was measured in Great Lakes sediment for these chemicals. So together, the data appears to say that the topographic barrier, this mountain peak, does influence the deposition, and you can certainly have this conveyor belt effect on either side that brings up pollutants from agricultural and urban areas. And so I would say in the historic use pesticides we saw, I think the fact that we don't see a decrease in concentration is due to continually plowing our fields, digging up those pesticides that are still in the soils, them revolatilizing and being transported to our parks. So one last part of my talk, does this really matter? People, you know, I mean, yes, I guess we tend to think that these mountains would be pristine, but you know, there's not many places that, well, there's nowhere I know that's pristine. So, but does it matter? Is it really affecting either human health or the health of wildlife? And so that's a fair question. We need to answer that. And so what we did was to measure the concentrations of those different pesticides in the fish we collected from those lakes, and then try to figure out, using the US EPA's own guidance on how to figure out if the concentrations in the fish could adversely impact the health of people eating the fish or increase their risk of cancer. And certainly there's huge health benefits to eating fish, from the lipids that are in the fish, from many factors of the fish. But one thing we have to consider is if there's an increased risk of cancer. So given the concentrations we measured in fish, we made some assumptions based on US EPA guidelines for different types of people that would be fishing in the lakes. Some of them would be just recreational fishers, maybe like you and I, eating at most 2.3 meals per month. Others might be native people, especially in Alaska, that could be going into lakes and eating up to 19 meals per month. So we did the calculations either way. So let's look at that data. So these are the concentrations of dieldrin in the fish from those different parks. Dieldrin is the orange bar. Para-DDE is a metabolite of DDT. And so when an organism is exposed to DDT, it will transform it in the body to DDE. So you often will measure DDE and not DDT. And so that's the green bar. It's orange and green. These are the different parks. Here's Sequoia down here. Romo is Rocky Mountain. Mora is Mount Rainier. Olympic, Glacier, Denali, Gates of the Arctic, and Noatak. So we're moving from south to north in this direction. And so we measure these chemicals in fish. It's on a log scale on the y-axis. Well, how does that relate to concentrations that have been measured in other areas? Well, salmon from the Pacific Ocean, this orange bar is the concentration of dieldrin in Pacific salmon. So we have some parks that are lower than that. Well, we have some parks that are definitely higher than that in terms of dieldrin. Other studies have been done in the Canadian Rockies. I should say that we collected and measured primarily trout, and so we could compare to these data to some degree. That's this red bar, and so our concentrations in general are higher than what were found in the Canadian Rockies. And of course, we have a lot more agriculture around our national parks than where these studies were done. So what about DDE concentrations? This bar is for salmon out of the Pacific Ocean, this green bar. This is for those Canadian mountain trout. So our DDE concentrations, some are, I guess, more, are lower than that bar. The top of the bar is the mean concentration. These individual circles are individual fish measured out of that lake. So it depends. You know, Sequoia has fairly high concentrations of both. Glacier also tend to have higher concentrations of both. And that's where the agriculture is. So is it relevant? So what about recreational fishers, like you and I, maybe only eating 2.3 meals per month out of a park? Here are the dieldrin concentrations for those different parks. This line is the line that US EPA would tell us anything below this is safe to eat. Anything above this, you would increase your risk of getting cancer by 1 in a million. So individual fish did surpass this line, but the mean of the different parks did not. So for a recreational fisher within these parks, not a concern. But the picture changes a little bit when we look at the 19.3 meals per month for the subsistence fishers. And so the green is dieldrin, the orange is dieldrin, the green is DDE, and the heptachloroepoxide, a form of the chlorines, is blue. And so the various lines then match to the various bars. And so we see, for example, for dieldrin, this is the US EPA line. These are the concentrations that would exceed the risk above it, not exceed the risk below it. So in parks like Sequoia and in Rocky Mountain National Park, and some individual fish in some of the Alaskan parks exceed those values. So when we talk about subsistence fishers or people who are in the parks eating larger amounts of fish from the park, and native peoples who would be using that as a resource, there could be a bit of a risk. So in some ways, it was surprising. So that brings me to the conclusions of my talk, and I hope you're also convinced I have one of the best jobs in the world for all of the wonderful places we get to measure. So I hope you see that SOCs, these semi-volatile organic compounds, undergo regional and trans-Pacific transport and deposition in the western US. This trans-Pacific transport plays somewhat of a role. It's episodic. It tends to be in the spring of the year, but it's not the main source. Even those historic use banned pesticides that are showing up are not from use in China. They're from our past use. They're from persistence in soil and continued revolatilization. Forest fires result in the release of these stored pesticides and PCBs. PAHs are released, of course, but we see that these forests are great at cleaning the atmosphere in a number of ways, and we can see re-release of those pollutants. Regional agricultural emissions of CUPs and HUPs, current use pesticides, historic use pesticides, definitely regional agriculture is important in that and bringing pollutants to mountains. And we saw in Rocky Mountain National Park that these topographic barriers influence the deposition in remote ecosystems. So very complex terrain, very complex deposition of pollutants. And we saw that, in fact, there was toxicological relevance, both for humans and for piscivorous wildlife. And I didn't talk about this here, but other researchers, Mike Kent and Carl Schreck at OSU, studied intersex in these same fish we measured and found some evidence that the male fish actually did have, in some cases, enhanced hormones that would suggest the formation of eggs, and in fact found some eggs formation in male fish. And we don't know if it's directly linked, but there's definitely potential risk to humans and for piscivorous wildlife in terms of the concentrations we measured. So I definitely need to acknowledge the students that I've had over time, and the students in the black bold are the students that have contributed directly to what I talked about today. And of course, we wouldn't be able to do what we do without funding from the National Science Foundation, primarily funding the atmospheric transport side, atmospheric chemistry side, the National Institutes of Health with our OSU Superfund research program studying those polycyclic aromatic hydrocarbons, and also studies in China, and the U.S. Department of Interior National Park Service for their funding of the national park work. And thank you for inviting me.